While Pia is preparing her presentation, I want to introduce her. Pia Valacco works at ICGEB, that is our colleagues in Buenos Aires from the mass spectrometry unit at the University of Buenos Aires. They are led by Silvia Moreno. I'm sure many of you here in Argentina have heard of Silvia Moreno and have sent her to ICGEB a lot of samples. And they did pioneer work in Argentina, starting proteomics work. They were the first unit in our country. Now, we are really happy to have Pia in representation of ICGEB here with us. And she's going to introduce to you the basic aspect of, oh, I hope nobody falls there. We're going to put the, yes. I know that all of you may have different levels of knowledge about mass spectrometry. So the first talks are going to try to level the knowledge in mass spectrometry. So the first part from Pia is to give a general introduction to the core concepts. If you have any questions, Pia is an expert in mass spectrometry for many years. So thank you, Pia, for being here.
Well, good morning, everyone. Thank you, Germán and Nico, for the invitation. I'm very happy to be here, very happy to see your faces directly after 2 years of Zoom presentations and lectures. It's really exciting to be able to see people personally. So we're going to start going through basic important concepts on mass spectrometry, and then in this first part of the lecture, we're going to understand how we identify proteins through mass spectrometry. If you have any questions, feel free to interrupt and ask whatever you want to ask, or to comment whatever you want to say. Okay, so the original definition of proteomics comes from an extrapolation of genomics, and it refers to any large-scale analysis of proteins. When we're studying a proteome, we're actually doing a complete study of the structure, localization, post-translational modifications, which we will now refer to as from now on as PTMs. We're studying functions and interactions of the proteins expressed by a cell, tissue, or organism. Since proteins and their PTMs can be studied by mass spectrometry, through the exact measurement of their masses, the everyday definition of proteomics is the study of proteins through mass spectrometry. When you're doing a mass spectrometry experiment, there are 3 main steps that you have to go through. So the first step is that you need to generate gas phase ions of the analyte. You need to get the analyte to ionize in the gas phase. We need to be able to separate those ions according to their M/Z. M/Z is their mass to charge ratio. Okay, this is something very important that we will be seeing over and over in proteomics. The analyte has a certain mass, it will ionize, and it will acquire a certain charge. So we will talk about the M/Z of the ions. Whatever has a charge will be detected in mass spectrometry. And then we need to be able to count those ions. We need to be able to measure them and see how many of those ions we have. That is why every mass spectrometer needs to have 3 basic components. We have the source, which is the place where the ions are generated. Then we have the analyzer, where we separate those ions. And we finally have the detector, where those ions impact and are measured. So every mass spectrometer needs to have at least one of each of these components. There are different types of sources, analyzers, and detectors that can be combined in different ways. Some instruments will even have more than one ion source, one analyzer, or detector. But at least you need to have one of each. For proteins and peptides, what we use are what we call soft ionization methods. Until the year 2002, there wasn't, you couldn't do proteomics because we didn't have these soft ionization methods. Ionization methods destroyed peptides. That's why they couldn't be analyzed by mass spectrometry. The soft ionization methods, there are two that are used for proteins and peptides. One is MALDI, which stands for matrix-assisted laser desorption ionization. In this method, what we do is we mix the sample with a matrix. They co-crystallize on a solid target plate by evaporation of the solvent. So then what we do is we shoot a laser on this mixture, and the matrix receives the energy of the laser and transfers it to the analyte, which will desorb and ionize. So that's why it's soft, because the matrix acts as an intermediate between the laser and the analyte. This energy transfer is pretty inefficient, and that is why in MALDI we usually get monocharged ions. You can work in positive mode or negative mode for any mass spectrometry experiment. That is, you regulate it by setting up the instrument and by changing solvents and matrices that you can use. For proteins and peptides, we usually use hydroxycinnamic acid or cinnamonic acid or DHB, which will help this ionization in a positive mode. And that's it on this. Then the other soft ionization method is SE, or electrospray ionization. In this case, the analyte will be dissolved in a liquid phase. It will go through a very fine capillary needle, which is under high voltage, and it will vaporize into fine multiply charged ion droplets. There is also high temperature and a nitrogen current that make these droplets become smaller and smaller until they evaporate and they release positive ions, if we're working in the positive mode, right? I'm talking about peptides already, so positive ions because we're working in positive mode. These ions are usually multiply charged. So if we want to compare a couple of advantages and disadvantages of MALDI and SE, we can say that the acquisition of spectra in MALDI is quick and simple because you just mix the analyte, the matrix, you let them dry, you shoot the laser, you get a spectrum. Then the analysis is long and work intensive for both methods. But the acquisition for MALDI is quick and simple. That's why it can be considered a high throughput technique. In the case of electrospray, a great, huge advantage is that since the analyte is in a liquid phase, it can be coupled to liquid chromatography. And this gives us a chance to work with complex mixtures. If in mass spectrometry, if you try to generate spectra from a very complex mixture, you will only, no matter how many proteins you have, you will only see the most abundant proteins because there's this phenomenon of covering up that the abundant proteins will cover up the signals of less abundant proteins. So if you can reduce the complexity of your sample through, for example, a liquid chromatography, you get a chance to dig deep down into the proteome. You will be able to detect less abundant proteins. So this is a very, very big advantage of electrospray. An advantage of MALDI is that the sample is stable after a lot of time. Like you can go back to a sample that you crystallized 3 months ago, you shoot it again, and you get excellent spectra because it's in a solid phase and it's very, very stable. In SE, once the sample goes through the electrospray needle, it is consumed. There's no going back to it. In MALDI, another advantage is that the spectra are easier to interpret. And now we're going to see a bit more about this. Since we have monocharged ions, usually, the spectra are simpler and they're easier to interpret. They're more straightforward. Now we'll see an example and you will understand it better. And an advantage of electrospray is that it's better for low molecular weight analytes. Why? Because we do not have a matrix that also ionizes and produces signals. In multi, the matrix will ionize and will generate signals in the spectra that may make it a bit dirtier, sometimes in the low molecular range area. So here are two spectra. The top one is a multi-spectrum. This one here is an SE spectrum of the same analyte. This is a pure protein RNAs of around 14 kilodaltons of molecular weight. So first of all, when we look at a spectrum on the x-axis, at a mass spectrum, on the x-axis we have the m/z value, the mass to charge value. And on the y-axis we have intensity in arbitrary units. This intensity, we will then see that it correlates with the relative abundance of an analyte in a sample, but it also depends on the ionization capabilities of the analyte. So if we look at this top spectrum, we see that it is very clean. It's very clear that we have a main peak around 14 of 14,000 m/z, and that corresponds to the +1 ion of the intact protein. We also have a little +2 here, which corresponds to the doubly charged. Proteins may sometimes acquire +2 or +3 charges since they are bigger. In peptides, it's very rare in multi to see more than +1. So we get a +2 at around 7,000, which would correspond to the doubly charged ion. In the case of the SE spectrum, we see that it's not as clean or as easy to interpret. We get a lot of signals, even though we're talking about a pure intact protein. And why is this? Because the different peaks correspond to differently charged species. So we have a +5, +6, +7, +8. So the determination of this charge is done by the instrument, by the software that controls the instrument. We will now see how it does it so that you understand it, but you usually won't have to do it. But once you get the charge and you have the m/z, you can calculate the molecular weight of the analytes. You multiply the m/z by the charge, you subtract the protons that were required for the ionization, and you get the molecular weight. But as you can see, it's not as simple to interpret on a first sight basis. Some important concepts that we need to understand. One of them is resolution. The resolution is the ability of a mass spectrometer to distinguish between adjacent ions with different mass to charge ratios. So here we have different spectra of the same analyte that have been acquired at different resolutions, so going from a lower resolution to a higher resolution. And you can see that here on the left, we get one very wide peak that appears to correspond to more than one species, but we can't really tell what we have in here. As we increase the resolution, we can see that the peaks become more defined, and we are able to determine their m/z's more clearly. So the better the resolution of an instrument, the more expensive it's going to be. And you can usually, within the resolution that your instrument has, you can decide to work at the best resolution, but this will also imply more time for acquisition of the data. So it's sometimes a compromise that you have to do to decide on resolution versus time. Here we have two classical ways to calculate resolution, but I don't want to waste time on this. I'm just leaving it here in case you want to check it. It's very simple, and the instrument does this. All you do is check that the resolution is okay when you're working. But here, this is pretty self-explanatory. Mass accuracy is another very important concept. It's the maximum error expected during the determination of molecular masses. So something that is very important to know is that all mass spectrometers are actually not very accurate. We are talking of high mass accuracy instruments, but they're only high mass accuracy instruments as long as you perform calibrations. Every time you are going to generate a spectra, you need to perform an external calibration of your instrument. What does this mean? What you're going to do is you're going to start your day by generating a spectrum of analytes of known masses. So once you generate that spectrum, you're going to tell the instrument, okay, this peak that you see over here, the real mass of this peak is whatever. And you're going to do that for all those peaks. So that's the way in which you calibrate your instrument, and that's the way in which your instrument may be considered a high mass accuracy instrument. Mass accuracy is usually expressed in percentage or parts per million. For example, if we have an instrument that has, for a mass of 1,000, has an error of 0.1 Dalton, what we say is that in parts per million, it's 100 parts per million, because if for 1 Dalton, the error is 0.1, for 1 million, it would be 100. So that's like a universal, simple way to express the error of an instrument. The error of an instrument is set by the vendor, by the factory that makes it, but then it also varies according to a lot of factors within the instrument. So every time when you calibrate, you get an idea of the error that you're working with that day or at that time. Sometimes when you clean the source, it will get better again, so it's something that varies a lot. Okay, more important concepts. Average and monoisotopic mass. As you all know, the elements in the periodic table, they have natural isotopes that have different relative abundances. So for example, hydrogen, we all know we have hydrogen 1 with a 99.985 relative abundance, but we also have hydrogen 2, deuterium, which has a relative abundance of 0.015. So actually, when we look at the periodic table and we look at the average atomic mass of an atom, what we have is a weighted average of all its isotopes. Okay, for example, for carbon, we have carbon 12 and carbon 13. So when we talk about the average mass of a molecule, what we are talking about is the addition of the average atomic masses of the atoms that compose it. High-resolution mass spectrometers are able to measure monoisotopic mass. One thing. When you have a molecule, of course, you are going to have molecules that have different combinations of the different isotopes for each atom. There is one of those combinations that is what we call the monoisotopic mass. So what is the monoisotopic mass? The monoisotopic mass is a molecule that is made up of only the most abundant atoms of each of the elements. So high-resolution mass spectrometers are able to distinguish between the masses of monoisotopic atoms or the different monoisotopic molecules or the different combinations of molecules. So for example, when you look at a peak like this one on this spectrum, and you zoom in this peak, what you actually see is a family of peaks. And what do these family members correspond to? They correspond to the different, what we call the isotope distribution pattern, the same molecule with different isotopic distributions. So the first one, which is the one with the smallest mass in each family, let's say in each group, is always going to be the monoisotopic one. It is the one that is made up of the most abundant isotope in each molecule. Then the next peak in the family is going to be the one that has all its atoms, which are the most abundant except for one. So one of them is going to be not one of the most abundant. So for example, it'll have all its hydrogen 1 atoms But the carbon atoms, all of them except for one, will be 12. But there is one that will be 13. So that is why there is a delta of 1 between these two peaks. Okay, so we know that the difference in mass between consecutive peaks in an isotope distribution pattern is 1. Okay, so it's important to know that the first peak is the monoisotopic one. But this is also very useful because it is used for charge determination. As I told you before, the instrument does the charge determination, but it is important to know how it does it. So how does it do it? For example, if we know that the m/z difference between this first peak and the second one, this is 685.5 and this is 685.8. So the difference in m/z is 0.3, right? And we know that the difference in mass is 1 because it has to be 1 every time. So we know that the charge of this peak is going to be 3, okay? So basically this difference in the m/z is going to tell us what the charge of the peak is. Here we have another example where the difference in m/z is 0.25. So the mass difference 1 divided 0.25 is going to give us a charge 4, okay? So this is the way in which we calculate the charge for a peak. And then it's important to remember that the first peak is always a monoisotopic, and then along the way on the isotope distribution pattern, we're going to somewhere have the average mass. But nowadays, we don't work with average mass anymore in proteomics because we have these high-resolution instruments that are always capable of distinguishing between all these peaks. Okay, so having gone over those basic concepts, we can now go into protein identification by mass spectrometry. So we're going to talk about two different approaches. One is the classical one. It's hardly ever used anymore, but it's very important conceptually to get to the next one. And it's also important that you know it. So we have two types of approaches. We have MS experiments or MSMS experiments, also called MS2 or Tandem mass spectrometry. In MS experiments, what we do is we're going to measure the exact mass of peptides derived from a protein. And that is how we're going to be able to identify a protein. In MSMS experiments, what we're going to do is we're going to measure the exact mass of the fragments obtained upon the fragmentation of a peptide. So we're going a step further. In the classic MS approach, which is also called peptide mass fingerprint, we start with a protein of interest or a mixture of proteins of interest. What we're going to do is we're going to digest them with a site-specific protease. It's very important that this protease is site-specific, that we know where it's going to cut. And now you're going to understand why. So we digest and we get a mixture of peptides derived from that protein. Those peptides go into the mass spectrometer and we generate a spectrum, okay? We said that we have m/z on the x-axis, intensity on the y-axis, and each peak in an MS spectrum corresponds to the m/z of a peptide, okay? Each peptide is going to generate a peak with a determined m/z. So how do we identify a protein from this? We have lots of specific softwares that are called search engines, that what they will basically do is they will do the same thing that we did on a wet lab. They will do it in silicon. So what we need is to tell the software exactly what we did. So we're going to get a database of known sequences. So if we're working with a protein with a certain sequence that we know, we're going to give the software that sequence. Or if it's cell culture extract from a human cell line, we're going to tell the search engine, okay, you have to digest the human proteome. We're going to tell the software which enzyme we used for the digestion so that it knows where to cut the proteins in the database. And a couple more things that we will see later, but basically the software will cut the database, generating a list of peptides, and then with that list of peptides, it will be able to generate a list of masses, okay? A list of expected theoretical masses that are derived from the digestion of that database. So once it has the theoretical masses, it can then extract the experimental masses from the spectrum, and it will compare both lists, generating a list of possible candidates. So one more word about the site-specific proteases that we use. There are many different enzymes that can be used. The most common one is trypsin. Trypsin cuts at the C-terminus of lysine and arginine. It is used because it has high cleavage specificity. It will cleave every lysine or arginine except those followed by proline. It's good because it produces peptides with C-terminal basic residues, which allows for good fragmentation series. We'll then see a little bit more about this. It can be modified to prevent autolysis, and it produces peptides between 80 and 20 amino acids in length because of the distribution of lysine and arginine generally in proteins. And this is pretty good for the mass range that we use for proteomics. It's not so important when we're working in electrospray because you can have bigger peptides that with multiple charges will end up in a smaller M/Z range. But in multi, when you usually have +1 peptides, it's good to have peptides within this mass range. So this green thing is a protein, and trypsin will cut in the yellow arrows where we have lysines or arginines, as long as they're not followed by proline. We can get, there's something that is important, is that you can sometimes get missed cleavage sites. So that is when the trypsin skips a site where it should cut. This can be due to basic or acidic residues adjacent to the cleavage site. It can be due to proline, or it can sometimes happen because your trypsin is a bit old. So it's important to know how your trypsin is working in order to be aware about these miscleavage sites that now we'll see why it's important to have them in mind. So this is an example of the form that you have to fill when you're going to do a search. You're not going to be seeing, there are many softwares that can be used for database searching. I'm showing Mass Spec because you're not going to be seeing it in the rest of the lectures, but they're all regarding what they ask you to fill in, they're all pretty similar. Then they all have different algorithms, different scoring systems, but regarding what they ask you to tell them, they're all pretty similar, and this one is straightforward, so it's easy to show you an example with this. So when you're going to do the search, once you have acquired your spectrum, you have your experimental peak list, and you need to do the search. So what do you have to tell the search engine so that it can perform the search? So basically, it's going to ask you, well, first here we have like some general bureaucratic information. Then it's going to tell you, okay, which database or repository do I get the database from? Then you have to tell the software which taxonomy you're working with. Most search engines now allow you to upload your proteome or database against which you want to search. Most of the paid ones. The free ones, maybe not so much. Well, no, but some of the free ones, you can upload it too. So you're going to have to tell the software which is the proteome that you're working with. You have to tell the software which enzyme you have digested with, how many miscleavages you want to allow. So if your trypsin is working well, and you know this because you usually digest standards every week, you're going to say, okay, let's allow 1 miscleavage, but if it's not working so well, you might go up to 2 or 3. Then you have to let the software know if you are expecting any modifications on the amino acids. So for example, in our case, and in most cases, before you perform the digestion, you treat your samples with DTT to reduce disulfide bridges, or not DTT or any reducing agent. So you open up the disulfide bridges, and then you want to block the cysteines so that you don't have folding again, right? So to block those cysteines, there are a couple different methods that you can use. In our case, we use iodoacetamide that will carbamidomethylate the cysteines, and it will generate an extra mass of 58 in each cysteine. So we have to let the software know this, and in our case, since we are using iodoacetamide in excess, we expect every cysteine in the sample to be modified. So we say, okay, let's consider carbamidomethylation a fixed modification. So every time the software sees a cysteine in the sequence, it will consider the mass of the cysteine plus the carbamidomethylation. So if I don't put this and I'm working with modified cysteines, I won't identify any peptide which has a modified cysteine because the masses will never match. So this is very, very important. Then we can also fill in variable modifications, and this means modifications that may not have affected all the, for example, all the cysteines or whatever amino acid I'm talking about. For example, we know that proteins, when they are stored, may get oxidized in their methionine. So we are expecting some methionines to be oxidized and some maybe they're not oxidized. So we say, okay, let's put oxidation of methionine as a variable modification. So some of them will be, when it comes across a peptide that has a methionine, it will calculate the mass with the plus 16 of the oxidation or without it. So the same thing for phosphorylation. If you're expecting phosphopeptides, you may say, okay, let's consider phosphorylation as a variable modification. Then we're going to have to give the software a peptide tolerance. So what is the allowed error that I want to work with between experimental masses and theoretical masses? So this is going to depend mainly on the instrument I'm using. I will know the mass accuracy of my instrument approximately, so I'm going to be working around that range. This you can fill in Daltons or in ppms, but it has to reflect the performance of the instrument you're acquiring your data with. Then this protein mass is optional and it's not really useful. Then you have to tell the search engine if the mass values you're giving it are positive ions, if you're working in the negative mode, or if you're giving it neutral values. If you're giving it monoisotopic masses or average masses, that also depends on your instrument and the software with which you generate the peak lists. And then finally, you're going to load your experimental data with which you want to compare the theoretical data that the search engine is going to generate. So this is just an example of how Mascot shows the results. It's a probability-based scoring, and it will give you a list of probable hits with a score and significance value. It's very important to bear in mind that hits in proteomics are always probable hits. We don't get real hits, okay? It's just a probability-based hit. So we get a list of proteins from the best to the worst, and a certain score, a p-value or expectation value, and then we will need to validate these candidates with other methods. So if we go in a bit further into the results form, what we get is a sequence coverage. This sequence coverage actually comes from the match of observed peptides and expected theoretical peptides. So basically, this was part of my experimental mass list. And all these masses belong to a protein in the database, which correspond to these peptides with these sequences. So basically, the software uses these sequences to generate the sequence coverage. But actually, we haven't seen any sequences in our results. All we've seen are mass lists that match theoretical mass lists that correspond to peptide sequences that belong to a certain protein. So that's the way in which we identify proteins by an MS approach. A couple of considerations to bear in mind. Although all the peptides in one protein are present in equimolar amounts, they do not have the same intensity in the spectrum. So one would expect that if you have a protein which produces 5 peptides, they're all in equimolar amounts. So if we say that intensity is correlates with relative abundance, we should get a spectrum with this pattern, right? But we don't. We get peptide spectrum with these kind of patterns. And why is this? Because, as I mentioned lightly before, peptides have different ionization capabilities. So even if they are in equal amounts, they will have different intensities due to their different ionization capabilities. For example, peptides with arginine in their C-terminus will give a stronger signal than those ending in lysine, because arginine has a stronger positive signal and better ionization capabilities. These ionization capabilities cannot really be predicted, so we know a couple of empirical data about it, but we can't really predict how well a peptide will ionize. And that is one of the main difficulties of why it is very difficult to do quantitative proteomics, okay? Why? Because mass spectrometry is really not a quantitative technique in itself. We have managed to get there with a lot of adaptations and approaches, but it's not quantitative by itself, because different analytes will ionize differently, so it's difficult to quantify them. And that is why when we do quantitation, we're actually quantifying the same peptide in different conditions, because that same peptide, we do expect it to ionize equally every time. Actually, it kind of can depend on the context too, but we don't usually take that into account. So when we quantify, we are quantifying the same peptide in different conditions, because it is very difficult to compare between different peptides. You can do it. You can normalize against a peptide that doesn't change in a sample, but you have to do like a lot of extra considerations to be able to do it. And then one more consideration is that the number of peptides identified for a protein depend on the amount or the mass of the protein analyzed. Okay, so for example, if I inject 500 nanograms of a protein, I might get, of a mix of a digested protein, I might get this spectrum with 5 peptides. But if I inject, I don't know, 1 microgram, I may get additional peptides that in this case were below the detection level. So the more you put of a protein, the more peptides you will be able to detect. But it is important that the relative intensity among those peaks will always be maintained. Okay, it will all go up, but the relative intensity will be maintained. Okay, now we go on to the other identification approach, which is tandem mass spectrometry, or MSMS, or MS2. In this case, the first part is just like what we saw before. We digest a protein, we generate the MS spectrum, okay? So we said that in this spectrum, each peak corresponds to the M over Z of a peptide. So what we're now gonna do is we're gonna select certain peaks. Which peaks are we gonna select? It depends. We can select the most intense peaks, the ones that are separated from the noise, the base noise that we have in a spectrum, peaks that are separated from their adjacent peaks, or we may want to decide on certain peaks that are of particular interest to us. So that will depend on your objective and your experimental design. But you select an ion, and you tell the mass spectrometer, okay, I want you to filter out the rest of the ions and just fragment this ion that I have selected. So basically, depending on the instrument, this is going to be different, but this is like a general scheme. All the ions will ionize, so you have a mixture of all your ions that ionize, and you will be able to filter out the rest of the ions so that only the molecules of the mass that you have selected for fragmentation will move on in the path of the mass spectrometer and get to the collision cell, well, to the fragmentation cell, because there are different fragmentation methods. So in this case, we call it a collision cell because we're going to fragment through collision-induced dissociation. So inside this fragmentation cell, the peptides will fragment by collision among the molecules of the peptide. You can insert an inert gas to make it better, and you will get all the fragments of the peptide. So once you the spectra that you will get is a spectra where now each peak corresponds to a fragment of the peptide that you selected. Okay, peptides may be cut along different points of their backbone, but when we use CID, they usually get cut at the peptidic bond. So we get what we call B ions if the charge is left on the N-terminus, or Y ions if the charge is on the C-terminus. So basically, how do we use this for protein identification? In the ideal situation, what you will get is that your peptide will fragment on different points. So here we expect the aspartic to be fragmented from the leucine. So we get these two fragments. Then in another molecule, we expect it to become fragmented here, or here, or here. So if you get a pretty complete fragmentation series of a peptide, what you can do then is you can calculate the difference between two consecutive ions. So between Y2 and Y3, the difference in mass between these two ions is the mass of amino acid C. Okay? So the difference between the masses of the consecutive fragments give us information on the amino acids that compose that sequence. So this is an example of de novo sequencing, which you won't have to do, but you can predict a sequence from a spectrum, from an MSMS spectrum. But what we usually do for protein identification by MSMS is we have search engines again that now what they will do is an extra step of in silico fragmentation. So the software will fragment all those peptides in the database that have the mass that we decided to fragment. So we will get theoretical fragmentation lists, and the software will do the match. So this is the form. It's basically the same, but we have an additional part where it asks for MSMS tolerance. So the mass accuracy for the MSMS acquisition, which is not always the same as the MS. And the information we get is the same as the one I showed you, the result form, but there is an additional part where we have these information about the MSMS spectrum. So we get the MSMS spectrum, it marks in red all the experimental peptides that match the theoretical ones, and then here we have, this is the predicted sequence, and in the table it shows all the theoretical peptide fragments expected, and it paints in red the ones that match the experimental ones. So to summarize, we can say that MS identification is based on the match between the experimental masses of the proteolytical peptides derived from a digested protein sample with the masses of the virtual peptides generated by theoretical digestion with the same enzyme of all the sequence contained in a database. In the case of MSMS identification, it is based on the comparison of the experimental fragmentation pattern of a tryptic peptide with the theoretical fragmentation of all the peptides of the same M/Z in the database. Both approaches are based on different criteria. To identify a protein with an MS approach, you need several peptides matching experimental M/Z. You can't identify a protein by MS with one peptide. You will need a group of peptides. To identify a protein with an MS/MS approach, a single spectra with a complete Y&B series may be enough. This is actually not true anymore because you need at least 2 or 3 unique peptides to validate a protein. And then the last thing I wanted to show you, I'm not going to go deep into this because you're going to see it in the afternoon, but these are 2 different mass spectrometers. They are the ones that we have at the ICGEB. And one of them is a MALDI-TOF-TOF. It's an Ultraflex 2 from Bruker, and it has the MALDI ion source with the laser. The analyzer is a TOF, which stands for time of flight. It will separate peptides based on their M/Z, and it will determine the mass based on their time of flight, on how long it takes for an ion to reach the detector. And then the other one is the AQX Active from Thermo Scientific. It has an electrospray source, and it is coupled to a nano LC. And it has a quadrupole for ion filtering or focusing, and it has an Orbitrap as a mass analyzer. Here, what will happen is that different ions will make a different orbit around this electrode, and according to that orbit, with a Fourier transformation, you can calculate the M/Z for the peptides. Well, and that's it. We can see this later in the next lecture, so we can stop here and then we go on. Thank you. I don't know if there are any questions, pero.
Sometimes when I read bibliography, I read the sentence data-dependent acquisition. What's that?
We're going to see that in 10 minutes, but data-dependent acquisition means that the acquisition of the selection, it's a very good question because I mentioned it very briefly without giving it a name. When the data-dependent acquisition means that you leave the instrument to decide based on the data, it will decide what it selects for fragmentation. So depending on the quality of a spectrum or of a peak in a spectrum, the instrument will decide if it chooses it for fragmentation or not. So now the next presentation starts with that. So in 10 minutes, if I don't answer it, you ask me again at the end of the next presentation.
Nice presentation. In the last picture, in the other picture, in MALDI-TOF-TOF, what is occur the collision fragmentation, the second mass transition? What is the collision cell?
Yes, good question. I didn't show it because I was running because I didn't have time. Actually, what in this instrument, what happened, the collision cell is like, it's above the TOF here in the first part, and when you decide to work in MS/MS mode, there is like an elevator that brings it down. So imagine that it would be like right here. So the thing is, when you work in MALDI, you first do an MS spectrum, so you shoot the sample, you generate the MS spectrum, you decide what you're going to fragment. So you say, okay, I will choose this peak because it's very intense and I want to fragment it. So you change the mode in the MALDI to the MS/MS mode. Then that is when the collision cell comes down. Actually, at one point in our lab, it was broken, so we had to like manually move it up and down. So it comes down, and you have to shoot again, so you're shooting the same sample again. The ions will all ionize because ionization, I mean, everything ionizes. And then before the collision cell, there is a gate that will act as a filter. So all the ions ionize, they start flying towards the collision cell, but this gate will only open at the time that the ion of the mass you told it to comes through. So the gate opens, the ion goes into the collision cell, it is fragmented, we inject, I think it's argon, yes, a noble gas, I think it's argon, so that we promote a better collision of the molecules, and then those fragments go on through the path of the TOF, and they reach the detector. But it's a good question because it wasn't here. The collision cell comes up and down, but it comes down at around this point. There is a very short time of flight for the ionized ions. They get filtered and they get fragmented there.
Thank you.
Germán, in the back.
I have two questions.
Mm-hmm.
The first question, what you consider to determine the resolution of the mass spectrometer when you're going to analyze a sample? And
Sorry,
the other
I didn't hear one of the words. What do you consider?
To determine the resolution of the mass spectrometer. And the other question is, how much is this technique limited by the database?
Okay, so for the first question, resolution, you will probably try to work with the best resolution you can with your instrument, but you have to consider time, also, because when you increase the resolution, so I have one slide that this will be useful here. So I just told you about how we work with the MALDI and how we shoot, we make an MS, and then we shoot again, and we make the MSMS. When you're working with, so in the MALDI, you will usually work with the best resolution possible. Sometimes, if it also depends maybe on the size of your analytes. In the case when we are sometimes working with intact proteins in MALDI, we might bring the resolution down a bit to get maybe a higher mass range, like the M/Z. If you work in high resolution mode, you can measure up to an M/Z of 5,000. So if you want to measure an intact protein, you may lower the resolution, but you get a bigger mass range, and you can get to M/Zs of 20 kilodaltons, or even 50 kilodaltons, or I mean 20 M/Zs or 50,000 M/Zs. But the resolution there will be much worse. Like you will get very wide peaks with zero resolution, but it's the only way to get to that mass range. That's the only way to measure masses of intact protein. So in the case of MALDI, it will be basically based on the size of the analyte that you're measuring. And then in the case of the Orbitrap, this is how the online LC-MS/MS works, how we make it work. So basically the Orbitrap, what it will do is it works in cycles. You have to remember that this is coupled to the liquid chromatography, right? So you have the peptides, they are in liquid phase, they go through the nano HPLC, and they elute from the HPLC, and they go directly into the electrospray needle. So they are ionizing constantly. It's going through all the time. So you set up a method where you tell your instrument to make an MS spectrum for every drop of the LC, and then you, if you're working in a data-dependent method, you say, okay, choose the 10 best peaks to fragment, right? And so this will take a certain amount of time because it's online, so you have to think that the sample is going through, it's ionizes here and then goes through here. You measure the MS. Once you have your MS, it makes a list of the 10 best peptides, and through to the next drop of the liquid chromatography, it will now select the first ion that it's going to fragment, and it will go to a collision cell, it will be fragmented, and then it will come to the Orbitrap to be measured, right? So all this has to happen in a certain amount of time that has to be compatible with your liquid chromatography, right? Because you have peptides eluting from the liquid chromatography, and they have to go through all these cycles. So the time that it takes will depend on the resolution with which you are working. So the higher the resolution, the longer it takes for the instrument to acquire the ions and in the collision cell, and to measure those ions in the orbit wrap. So basically, it will mainly depend on that, on the time frame that you have. The instrument time is the most precious time we have, the most precious thing we have in a mass spectrometry lab, because all the other things you can speed up, but the time that a sample is in the instrument is our bottleneck. That's where we get blocked. So you have to find a compromise between resolution and the time. Here, if you are working, I can see it there, at a resolution, I think it is 70,000 for the MS and 17,000 for the MSMS. This cycle of 1 MS and 10 MSMS takes 1 second, okay? Which looks like very little, especially if you're doing like a 2-hour run. But the thing is that you need to acquire many, many MSMS spectra to be able to identify thousands of proteins, which is what we go for when we have a complex sample. So if you're working at these two resolutions, each cycle will take 1 second, and after a 2-hour run, you can get to 60,000 MSMS spectra, which is something which is not even excellent for protein identification of a complex sample such as a cell culture extract. So it will basically depend on the time that you want to dedicate to measuring that sample. And then your second question was, sorry, I forgot. Well, it's very limited because everything I told you about database searching is 100% dependent on having the sequence or the proteome of the biological model you are working with. There is an alternative to not having the database, which is de novo sequencing. I went through it quickly because I also didn't have time, but what you can do when you're doing de novo sequencing is basically try to interpret a spectrum from scratch. So basically you have to try to identify the Y ions and the B ions in a spectrum. So you say, okay, this looks like, there are many rules and techniques and tips to be able to do it. Like people who do it all the time are pretty able at it, but you have to find like clue ions in the spectrum and then try to find the difference in mass between consecutive peaks and try to predict the sequence from scratch. But it's very, very difficult, very labor intensive, like it will take one spectrum, might take you 4 hours to do, and then you need to have some kind of at least a related database so that you say, okay, I found a 5 amino acid sequence, I have no idea what this is, so you do a homology search, and like with other similar organisms, maybe you get the complete sequence of, I don't know, transaldolasa 4, and you say, okay, this could be it, and then you get inspired by that, and you try to follow a bit more on the sequence. But it's very, the database is very, very limiting, and it's very difficult to do proteomics when you have no database with to search with. Actually, you can only do this with like multi-MSMS spectrum, because if in an orbit wrap you get, as I told you, 60,000 MSMS spectra, you're not going to be able to do anything manually with them. I mean, you could choose 5 and try to interpret them, but it's not going to be something that you can profit from. So when you don't have a database, you might try to do like some de novo sequence manually, or you need to find a close lineage of another species in order to try to do some kind of database searching. So it's absolutely limiting, I would say.
Thank you, Pia. One last question before the first break. What would be your advice for the audience, for those who are working with membrane proteins and in the transmembrane regions, you don't have many lysine and arginines. So what is the approach take when you have these kind of samples?
Yes, like let me show you. I think I have it here. So what we do when we have like complicated proteins, here, for example, here I didn't show this because also no time, but trypsin is not always the best choice. So this is an example of a protein that will get nicely digested with trypsin. But then, for example, this other protein, it's called P8, and the composition of lysis and arginines is pretty horrible. Because you get like very large peptides or parts that get chunked up and you can't see anything. So basically what you have to do when you have these kind of things is try to do parallel digestions with other kinds of enzymes. We have a battery of enzymes in the lab, and we usually, like, if you're doing a general thing that doesn't involve membrane proteins or doesn't involve strange proteins like the one I just showed you, you go for trypsin, and you're supposed to get like a global coverage of the general proteins. But if not, you have to start doing, and maybe more than one enzyme, like you digest part of your sample with chymotrypsin or glu-C, so they will cut at different places and you will be getting different peptides that may ionize better. So trypsin is not always the best choice. It's the one we use for general global approaches, but it's not always the one that you have to go for. So that's why it's very important. And I think that the message after this course, and usually after every proteomics course, is it's very important to focus on your experimental design before you start anything related to proteomics. And this implies a very fluent communication with the facility or the lab, which is going to be in charge of the proteomics analysis. Because as Eduardo will tell you in the afternoon, if your sample is not properly prepared, then the whole thing won't work. If the design is not planned correctly, it won't work. So it's very important to talk with us fluently before you start. And these courses are also great for that, right? So that you have a basic idea of where to start and how to target it.
Thank you, Pia, again. So we next move to our first break. The coffee breaks are going to be outside this room, so we wait for you to have a cup of coffee. We will return in a little less than half an hour. Thank you.